Hi, my name is Huyghe, and today I'll be presenting my project on investigating insecticide resistance in the invasive Culex pipiens mosquito. Culex pipiens is a great vector for diseases like West Nile virus in Canada. A vector is an organism or object that transmits an infectious agent from one host to another. Here is a transmission cycle for West Nile virus, where an infected bird or mosquito will transmit the virus to the other, and then the infected mosquito will transmit the virus to a healthy mammal. This is the mosquito that we're focusing on for this project, Culex pipiens, also known as the common house mosquito. They're invasive to Alberta and an important vector for pathogens like West Nile virus due to them being attracted to birds and mammals. They use a variety of environments and they're well adapted to urban areas because they use containers like water barrels for easy dispersal. They're also multivoltine, which means they produce multiple generations within a single year. Here's their habitat distribution, where it stretches from coast to coast of the United States, as well as into eastern Canada and recently western Canada, being found in Edmonton in 2018 and Calgary in 2022. Here's their life cycle, which starts with an adult female laying egg rafts, which are large clumps of eggs in stagnant water. These eggs hatch within 2 days, and then the larvae emerge and begin feeding on organic matter. They then go into the pupal stage and then the adult stage, where they engage in mating soon after molting and seek out sources of blood for nutrient acquisition necessary for egg production. This stage lasts for a few weeks. Some ways to control these mosquitoes are through insecticides like adulticides or larvicides. Some common adulticides are pyrethroids and organophosphates, which target the nervous system and cause paralysis in the mosquito. Larvicides work differently, with bacterial larvicides being ingested and releasing toxins in the mosquito after being placed in the water where they are. Growth regulators disrupt hormone balance, and oils or films can be placed on the surface of water to suffocate the mosquitoes. A problem with insecticides is that they can cause resistance. Knockdown resistance is a mutation occurring in genes of these mosquitoes that causes resistance to insecticides that cause paralysis, which makes it a problem for the adulticides. There have been trends of Culex pipiens being found to have this resistance in North Africa and Europe, and they are found in different genes like the voltage-gated sodium channel gene and the ACE1 gene, which are immune system genes. This leads me to the hypothesis that if the Culex pipiens genome contains knockdown-resistant variants, resistance will be present. We do this through these objectives of collecting genomes from public databases and lab samples, identifying known resistance mutations from the literature, and using a bioinformatic pipeline to evaluate if Culex pipiens have knockdown resistance identified in the literature. The methods we use are to conduct fieldwork to collect mosquito samples, taking these mosquitoes and sequencing their genomes, and then using these in bioinformatics software to operate through multiple steps of a pipeline and compare these to the public database samples. Here is the pipeline that we use, where we start with the raw reads. We go through quality control and trimming, mapping it to the genome, extracting the genes, generating sequences, aligning each gene, and identifying codon positions and resistance locations. These are the results that we found, where no knockdown resistance was present in the voltage-gated sodium channel gene or the ACE1 genes of Alberta Culex pipiens samples. Here on the left, we have the reverse complement of one of the VGSC mutation sites, where a TTT phenylalanine codon indicates resistance and a TTA leucine codon represents susceptibility. We see here that the leucine amino acid is present instead of the phenylalanine, indicating susceptibility. On the right, we have the ACE1 gene site, where a susceptible result is a glycine GGC codon and a resistant result is a serine AGC codon. Here we can see susceptibility. The numbers on the side each represent an individual mosquito sample from either Red Deer, Calgary, or Edmonton, for a total of 36 sample sequences, and not all can be seen here. Each one had the exact same result for these mutations, as well as others from the VGSC gene. For future steps of this research, we would need to evaluate resistance to BTI, which is a bacterial insecticide used in Calgary and other parts of the world, and resistance to pyrethroids with bottle assays. Here are my references, and I would like to thank my supervisors, everyone in my lab, and all of my collaborators for their help on this.